The overall goal of this protocol is to use state-of-the-art immunohistochemistry to accurately and rapidly identify skeletal muscle fiber types.
This method can help answer key questions in the field of muscle biology, such as whether fiber type changes are precipitated by loss of functional proteins. Though this method can provide insight into many models of muscle disease, it can also be used in systems such as knock-in and knockout models to reveal the greater impact of a single molecule on basic muscle function.
To begin, air dry frozen sections of mouse muscle on charged slides for about half an hour. Once dried, draw a border around the sections on each slide using a hydrophobic barrier PAP pen. Next, to block nonspecific antibody binding, load about 250 microliters of 5% BSA in PBS within the drawn borders and incubate the slides at room temperature for an hour. Meanwhile, prepare a 1 to 50 dilution of the primary antibody in 5% BSA PBS. Make about 250 microliters per slide and store the antibody dilution on ice. After the block period, aspirate the block solution from the slides and add 250 microliter aliquots of the antibody dilution. For a negative control, apply 5% BSA in PBS without primary antibody. Next, load a petri dish with dampened filter paper and cover it in aluminum foil for an incubation chamber. Then, incubate the slides at 4 degrees Celsius for 24 to 48 hours in the chamber or in a similar environment with the same humidity.